Okay, let's unpack this. Imagine a true botanical detective story, a groundbreaking innovation so significant it didn't just help plants adapt. It completely reshaped Earth's ancient landscapes and laid the foundation for the magnificent forests we still marvel at today. We're talking about the incredible evolution of the seed.
And our mission in this deep dive is to trace that incredible journey. We'll explore the ancient ancestors of today's dominant seed plants, then meet the diverse living groups of gymnosperms, literally the naked seed plants, understanding their unique features, life cycles, and the brilliant evolutionary strategies that allowed them to thrive. We're pulling back the curtain on this pivotal chapter in plant evolution, distilling it all for you directly from the latest research in Raven biology of plants.
Here's the key takeaway right up front, and it's a big one. The seed is a true marvel of survival. Think of it as nature's ultimate survival kit for an embryo. It offers unparalleled protection, shielding that tiny, vulnerable developing plant, and crucially, it comes pre-packed with its own stored food supply for those critical early stages of germination. This seemingly simple, yet profoundly impactful innovation is the principal reason seed plants have achieved such dominance globally. It gave them a massive selective advantage over their spore-shedding relatives. Now, you might wonder, what exactly makes a seed so revolutionary? It's far more than just a sturdy little casing, right?
Absolutely. The foundational concept starts with what we call heterospory. Unlike plants that produce just one type of spore, seed plants produce two distinct kinds. Megaspores, which are the larger spores destined to become the female reproductive parts, and microspores, the smaller ones that develop into the male reproductive parts. Now, while some seedless plants also develop this trick, the seed plants took it to an extreme, evolving a completely new structure for the megaspore, the ovule.
The ovule, right.
Essentially, a seed is just a matured ovule that now contains an embryo. The immature ovule itself is a marvel. Imagine a central fleshy tissue called the megasporangium, where those crucial megaspores are produced. This megasporangium is then enveloped by one or two protective layers, like a shield called integuments, leaving just a tiny opening at the top, which we call the micropyle.
Okay, so it's not just a spore floating off into the wind anymore. This sounds like a highly engineered package. And our sources detail 7 critical evolutionary events that, step by step, led to this brilliant ovule. It's like discovering the engineering blueprints for plant success. The first big step was the retention of megaspores within that megasporangium. No more shedding spores. That fleshy megasporangium in seed plants becomes what's called the nucellus, keeping its precious cargo safely tucked away.
That's right. And this retention triggered a cascade of other vital changes. Second, there was a drastic reduction. Only one megaspore mother cell would form per megasporangium.
Just one.
Just one. Third, out of the 4 megaspores the single cell produced, only one actually survived and became functional.
Concentrating resources.
Exactly. Fourth, the female gametophyte then developed entirely inside that single functional megaspore, what we call endosporic development. It's no longer a free-living, vulnerable entity floating around.
That's a huge shift in protection. What came next?
Fifth, the young plant embryo, the sporophyte, began developing within this protected female gametophyte, which itself was still safely encased inside the megasporangium. Sixth, those protective integuments we mentioned earlier formed, completely surrounding the megasporangium, leaving only that tiny micropyle opening.
The little doorway.
Precisely. And finally, seventh, the apex of the megasporangium evolved to specifically receive microspores, which, by this point, had evolved into what we recognize as pollen grains.
If we connect this to the bigger picture, these 7 steps, taken together, represent a profound pivot in how plants disperse themselves. They went from relying on delicate, exposed spores to a robust, self-contained survival pod, an embryo-carrying seed ready for the challenges of the terrestrial world.
It's a true innovation in packaging and delivery, and we can actually see glimpses of this evolutionary journey in the fossil record. The oldest known ovules, dating back about 365 million years to the Late Devonian, reveal these early stages. Take Elkinsia polymorpha, for example. Its ovule had a central nucellus, but its integuments weren't fully fused. There are more like 4 or 5 separate lobes curling inwards, almost surrounded by a separate set of sterile branching structures called cupules.
So you can literally visualize the evolution of these integuments as a gradual fusion process, almost like fingers fluidly zipping up around a precious core.
Exactly. It's a great analogy. We see fossil examples like Gymnospermic which had 8 entirely separate finger-like projections. Then Gymnospermalatans showed these lobes fused for about a third of their length. By Eurostoma angulare, fusion was nearly complete, and finally in Stamnostoma hutnens, the integuments were fully fused, leaving only the micropyle open. This zipping up was key to the seed's ultimate protection.
So, to quickly summarize the ultimate survival package, a mature seed today contains three main components: a miniature plant, the embryo, its packed lunch, the stored food, which in gymnosperms comes from the megagametophyte, that female part, and its durable casing, the seed coat, developed from those zipping up integuments. This incredible development begs the question, where did this sophisticated seed innovation even come from? What were its deep, deep ancestors like?
This takes us to the progymnosperms, a fascinating group from the late Paleozoic era about 290 million years ago. They're considered the likely evolutionary link between earlier spore-producing vascular plants, like the trimerophytes, and the seed plants. What makes them so special is their hybrid nature. They still reproduced using freely dispersed spores, just like their seedless relatives, but crucially, they also produced true wood, what we call secondary xylem and secondary phloem, the sugar-carrying tissue.
Wood before seeds.
Yes. And their most pivotal evolutionary advance, arguably, was the bifacial vascular cambium. Imagine a central growth engine in the stem, like a two-way street, constantly producing wood inwards for structural support and nutrient-carrying phloem outwards. This innovation, now characteristic of all modern seed plants, originated in these progymnosperms.
That's a huge step towards building truly enormous plants, and our sources highlight Archaeopteris-type progymnosperms as a major player. These appeared around 370 million years ago in the Devonian, forming Earth's very first extensive forests. Some Archaeopteris trees, fossilized as Calixylon, were massive, over a meter in diameter and 10 meters tall, creating these ancient towering woodlands. They already had a well-organized vascular system, a eustyl, where vascular tissues are arranged in a ring, much like modern seed plants. The truly insightful takeaway here is that some Archaeopteris were already heterosporous, meaning the ability to produce wood and different types of spores actually predates the evolution of the seed itself.
Fascinating. Absolutely. And beyond these early progenitors, there were other fascinating extinct gymnosperms. We have the seed ferns, or Pteridospermales, a hugely diverse group that emerged from the Devonian and persisted until the Jurassic. They ranged in form from slender, branched plants to tree-like structures, and while they looked like ferns, they bore actual seeds.
Wow, fern-like plants with seeds.
Yeah. Then there were the cycadeoids, or Bennettitales, prevalent in the Mesozoic, with palm-like leaves that superficially resembled living cycads. What's truly intriguing about some Bennettitales is their flower-like bisexual reproductive structures. This initially made some botanists wonder if they were direct ancestors to flowering plants. However, molecular evidence now tells us that was likely an independent evolutionary pathway, a kind of biological convergence. Interesting, but not the direct line to flowers.
Right, parallel evolution. Okay, now let's fast forward to the living gymnosperms we see today. The name gymnosperm itself literally means naked seed, right?
It does, and it perfectly encapsulates their defining feature. Their ovules and seeds are exposed, not enclosed within a fruit as they are in flowering plants. They're typically found on the surface of modified leaves called sporophylls.
Naked seeds out in the open.
That's right. Today we recognize 4 main living phyla. Conifer phyta, which are our familiar conifers, Cycadeoids, the cycads, Ginkgo phyta, represented by the singular maidenhair tree, and Nettophyta, the nettophytes. The exact evolutionary relationships among these 4 groups are still a bit of a detective story themselves, with ongoing debates among botanists, leading to fascinating hypotheses like the netifer and nepine theories about where nettophytes fit into the conifer family tree. It's still being actively researched.
One of the major breakthroughs that allowed seed plants to conquer land was their incredible solution for reproduction without needing external water. This was a game changer.
Absolutely revolutionary. In seedless vascular plants, their flagellated sperm need a film of water to swim to the egg. But gymnosperms found a different path. The partly developed male reproductive part, the pollen grain, is transferred directly to the vicinity of an ovule, a process we call pollination. After pollination, this pollen grain produces a pollen tube, a tubular outgrowth that carries the sperm. Interestingly, the microgametophytes of seed plants don't form the antheridia, those sperm-producing structures we see in seedless plants.
So the pollen tube takes over that delivery job.
Well, it gets interesting. What's fascinating here is a transitional stage we observe in cycads and ginkgo. Their pollen tube initially acts like a tiny absorbing root, a haustorial structure, digesting nutrients from the ovule tissue for months. Then it bursts open, releasing a few large multiflagellated swimming sperm, which then swim a short distance to the egg.
Swimming sperm, still.
Still, but only a very short distance. This suggests the pollen tube's initial purpose may have been primarily nutrient absorption.
So this little tube evolved from being a food gatherer to a sperm delivery system in other groups. That makes so much sense.
Precisely. In conifers, neophytes, and later angiosperms, the sperm are non-motile. The pollen tube evolved to directly convey these sperm right to the doorstep of the egg cell. This raises an important question. This evolutionary shift suggests the pollen tube's primary role evolved from nutrient uptake in earlier forms to a direct conduit for sperm delivery in more advanced ones.
And what about polyembryony? I remember reading about that.
Yes, a common feature across gymnosperms. The female reproductive structure, the megagametophyte, often produces several eggs within structures called archegonia. This means multiple eggs can be fertilized, and several embryos may start to develop within a single ovule. However, usually only one ultimately matures into a viable seed. It's like having multiple backups.
A bit of internal competition. Okay, let's unpack the most numerous, widespread, and ecologically dominant group, the conifers. These are truly the giants of the forest.
Indeed. The conifer phyta boast about 70 genera and 630 species, and they play a massive ecological and commercial role, often dominating vast regions, especially in the northern temperate zones. This group includes the tallest known vascular plant, the majestic redwood, Sequoia sempervirens, which can soar to over 115 meters. Just incredible trees.
Yeah, among conifers, the pines, genus Pinus, are probably the most familiar. Their leaf arrangement is really unique. Pine seedlings start with single needles, but adult pines typically arrange their needles in tight bundles called fascicles. It's like a little prepackaged bouquet of needles, isn't it? With a specific number, 1 to 8, depending on the species.
Exactly, a determinate short shoot. And these needles are also incredibly adapted for dry conditions. Think about it. A thick, waxy cuticle, a reinforced layer called the hypodermis, sunken stomata, those tiny pores for gas exchange are tucked away, and even resin ducts that act as a defense system against insects and pathogens.
Built tough.
Very tough. Their stems, of course, develop into substantial wood through secondary growth, primarily made up of tracheids for water transport and support. And their outer bark, the periderm, eventually replaces the initial epidermis for protection as the trunk thickens.
Okay, so how does reproduction work in pines? The pine life cycle sounds complex.
It is quite a journey, typically spanning 2 full years. On the same tree, you'll find 2 types of cones. The smaller pollen cones usually clustered on lower branches, and the larger ovulate cones typically on upper branches.
Why the different locations?
That strategic placement encourages cross-pollination by wind. Pollen from the lower branches is less likely to land directly on the ovulate cones of the same tree. Smart design.
Makes sense.
The pollen cones have scales called microsporophylls, each with 2 microsporangia where pollen develops. Microsporocytes undergo meiosis, producing haploid microspores. Each microspore develops into a winged pollen grain, which is actually the immature male gametophyte, containing just 4 cells at this stage. Then huge amounts of this pollen are shed in the spring.
So when that pollen is released, how does it find its target in the ovulate cones?
In the spring of year 1, the scales of the young ovulate cone are slightly open. The ovulate cone is a more complex structure, with ovuliferous scales each bearing 2 ovules, plus a little sterile bract. Inside each ovule is the megasporangium, the nucellus, containing a single megasporocyte that undergoes meiosis. Only one of the resulting 4 megaspores survives. As pollen grains drift by, some get caught between the cone scales and adhere to sticky pollination drops exuding from the ovule's micropyle. This sticky drop then contracts, pulling the pollen right into contact with the nucellus inside the ovule. Then the cone scales close up tightly for protection.
Like closing the door after the guest arrives.
Exactly. The pollen germinates and begins to form a pollen tube, but the female gametophyte development inside is incredibly slow. It takes about 15 months until the archegoniae containing the eggs are actually ready. Meanwhile, the pollen tube slowly digests its way through the nucellus towards the developing archegonia. During this time, the generative cell within the pollen divides to form 2 sperm cells. No antheridia are formed.
15 months to fertilization. That's quite a wait. Must be a record for delayed gratification in the plant world.
It certainly requires patience. Fertilization finally happens in the spring of the 2nd year, roughly 15 months after pollination first occurred. The pollen tube reaches an egg cell within an archegonium, penetrates it, and discharges its two sperm. One sperm fuses with the egg nucleus, forming the zygote, the beginning of the new embryo. The other sperm usually just degenerates.
And you mentioned polyembryony is common here too.
Yes, very common in pines. Since there are usually multiple archegonia, several can get fertilized, meaning several embryos might start developing within the same ovule. But typically, resource competition means only one fully matures. The developing embryo, a miniature plant with its first root, radicle, shoot apex, and seed leaves, cotyledons, is pushed deep into the stored food tissue by special elongated cells called suspensors. While all this is happening, the integument matures into that protective seed coat.
And what's truly remarkable about the pine seed itself is that it's a multi-generational package, isn't it?
Yeah.
Containing parts from 3 different generations.
Exactly. It's amazing. You have the old diploid sporophyte generation represented by the seed coat and any remnants of the nucellus. You have the haploid female gametophyte generation providing the stored food. And then you have the new diploid sporophyte generation as the embryo itself. A complete lineage in one tiny, robust package.
The time capsule.
A perfect way to put it. Most pine seeds have delicate wings derived from the cone scale and are wind dispersed when the cones open in the autumn of the second year. But there are brilliant variations. Think of lodgepole pines, whose cones are serotinous. They only open and release seeds after the intense heat of a forest fire. A true adaptation to their fire-prone environment.
Whoa.
Or consider pinyon pines. With their wingless, highly nutritious seeds, they rely on birds, especially Clark's nutcrackers, for dispersal. The birds harvest the seeds, cache them underground for later, and inevitably forget some, effectively planting new trees. A fantastic mutualism.
Beyond pines, the conifer family is vast. We have firs, larches, which are actually deciduous conifers, unusually spruces, hemlocks, and Douglas firs. They're all part of the Pinaceae family.
That's right. Then there are cypresses, junipers, and even the giant sequoias and coastal redwoods, now grouped in the Cupressaceae family. And the yews in the Taxaceae family are distinct because they produce solitary ovules encased in a fleshy, often brightly colored, cup-like structure called an aril. It looks a bit like a berry, attracting birds for dispersal, but botanically, it's not a true fruit.
So many familiar trees.
And we find some truly unique conifers globally, too. The Archaeopteris family, for instance, mostly found in the southern hemisphere, is an ancient lineage. It includes the incredibly rare Wollemia nobilis, the Wollemi pine.
The dinosaur tree, right?
That's the one. Only discovered in Australia in 1994, with fewer than 40 adult trees known in the wild. It's arguably the world's rarest plant species, a genuine relic.
Incredible find.
Then there are the giants, coastal redwood, Sequoia sempervirens, the tallest living plant, and Sequoiadendron giganteum, the big tree known for its immense girth, both native to California and now placed within the Cupressaceae. And let's not forget the dawn redwood, Metasequoia.
Another living fossil.
Exactly. It was widespread across the northern hemisphere millions of years ago, known only from fossils and thought long extinct. Then, remarkably, it was rediscovered alive in a remote valley in China in the 1940s. Now it's widely cultivated due to its beauty and resilience.
Moving beyond the towering conifers, we find some truly ancient and unique gymnosperms that, at first glance, barely resemble each other at all. It's like stepping back even further in time.
They certainly are diverse, almost like evolutionary experiments. First, we have phylum Cycadeoids, the cycads. These palm-like plants are predominantly tropical and subtropical, and they truly flourished back in the Mesozoic era, earning it the nickname Age of Cycads and Dinosaurs. They often have a characteristic look, a cluster of large, pinnately compound leaves atop a thick, often unbranched trunk with very slow secondary growth and a large pith. Be warned, though, many parts are highly toxic if ingested.
Good to know. What about those special roots you mentioned earlier?
Ah, yes, they're unique coralloid roots. These specialized roots often grow upwards, sometimes even emerging above ground, and they form a symbiotic relationship with nitrogen-fixing cyanobacteria, usually species of Anabana. This partnership allows cycads to thrive in nutrient-poor soils by getting a direct supply of usable nitrogen. Pretty clever.
So they've got this special underground or sometimes above ground relationship going on. And how do they reproduce? Differently from conifers.
Quite differently in some ways. Cycads are strictly dioecious, meaning you have entirely separate male plants producing pollen cones and female plants producing usually much larger ovulate cones. Their pollen tubes, like we touched on, initially act like those early hostorial structures, digesting nucellar tissue for months. Eventually, they rupture near the archegonia and release large, multiflagellated swimming sperm into a fluid-filled chamber created within the ovule. The sperm then swim that short distance to fertilize the eggs.
Swimming sperm again. And what about pollination? Wind.
While wind might play a minor role, cycads are overwhelmingly insect-pollinated. Beetles, especially weevils, are attracted to the cones, often by heat or specific scents, and they inadvertently transfer pollen as they feed or seek shelter. This relationship between cycads and insects is ancient, stretching back millions of years.
Fascinating. Next, we meet the extraordinary phylum Gametophyta, represented by just one living species, Ginkgo biloba, the maidenhair tree. It's instantly recognizable by its unique fan-shaped leaves with dichotomous venation. The veins fork repeatedly into two equal branches.
And here's where it gets really interesting. Unlike almost all other gymnosperms, ginkgo is deciduous, shedding its beautiful golden leaves every autumn. It's the sole living survivor of its entire phylum, a genuine living fossil, literally saved from extinction because it was cultivated for centuries in temple gardens in China and Japan.
Wow, saved by cultivation.
Exactly. Its incredible resistance to air pollution has also made it a popular and remarkably resilient urban street tree worldwide.
How does it reproduce? Similar to cycad.
Like cycads, ginkgo is also strictly dioecious, separate male and female trees. The male trees produce small, catkin-like pollen strobili. The female trees produce ovules, usually in pairs at the ends of short stalks. These ovules develop into fleshy-coated seeds in autumn, which are famous, or maybe infamous, for a rather foul odor as the fleshy outer layer decays, think rancid butter, thanks to compounds like butanoic and hexanoic acids. This is why male trees, which don't produce seeds, are strongly preferred for urban planting.
Understandable.
However, the kernel inside that smelly seed coat, the actual seed, is considered a delicacy in some Asian cuisines after proper preparation. And remarkably, fertilization involving those large swimming sperm released from the hostorial pollen tube can even occur after the ovules have fallen from the parent tree to the ground.
Fertilization on the ground. That's wild.
It is. And one more little quirk mentioned in the text. A Cuckomixa-like green alga is sometimes found living within the cells of ginkgo tissues, though its exact role isn't fully clear.
And finally, we arrive at phylum Bennettitales, home to three highly unusual genera that consistently challenge our botanical expectations of gymnosperms. They seem almost ungymnosperm-like in some ways.
Indeed. They are a puzzle. These are Needham, Ephedra, and Welwitschia. If we connect this to the bigger picture, these plants exhibit some truly surprising angiosperm-like features, even though current molecular evidence suggests they evolved these characteristics independently, not as direct relatives of flowering plants. Needham species are mostly tropical trees and climbing vines with broad, leathery leaves that have netted venation. Visually, they could easily be mistaken for eudicot flowering plants. Ephedra, on the other hand, consists of profusely branched shrubs with small, scale-like leaves, often found in arid or desert regions worldwide. Superficially, they might remind you a bit of Equisetum, the horsetails. Some Ephedra species are the source of the stimulant ephedrine.
And then there's Welwitschia mirabilis, easily the weirdest of the bunch.
Welwitschia mirabilis, often called the most bizarre vascular plant on Earth, and for good reason. Most of its squat, woody stem remains buried in the sandy soil of the Namib Desert. It produces only two enormous strap-shaped leaves throughout its entire, incredibly long life, potentially over 1,500 years. These two leaves grow continuously from their base and split lengthwise over time, becoming frayed and torn at the ends, making it look like it has many leaves. You'll find it exclusively in the coastal deserts of southwestern Africa, absorbing moisture from fog.
Just two leaves its whole life. Incredible. What about those angiosperm-like features?
Right. Well, their strobili, or cones, are often compound and can resemble primitive flower clusters. More significantly, their xylem contains vessel elements for water transport, in addition to tracheids. Among living seed plants, vessels were long thought unique to angiosperms, but neophytes have them too, a case of convergent evolution. Also, Needham and Welwitschia lack archegonia in their female gametophytes, another feature shared with angiosperms but different from other gymnosperms.
And this raises that incredibly important question that once really blew the lines: double fertilization. It was once thought to be exclusively unique to flowering plants, right? But it actually occurs in Ephedra and in Needham too.
That's a crucial insight, and yes, it caused quite a stir. In these neophytes, two sperm nuclei from a single pollen tube fuse with separate nuclei within the female gametophyte. One sperm fertilizes the egg nucleus, forming the zygote. The second sperm fuses with another nucleus in the female gametophyte. However, here's the key difference from angiosperms. In neophytes, this second fertilization event typically results in an extra embryo, which usually aborts early on. It does not form the distinctive, often triploid, nutritive tissue called endosperm that is the hallmark of double fertilization in flowering plants. So similar process, different outcome.
Similar mechanism, different product. Fascinating distinction.
And one more thing. Many neophytes also produce pollination drops that function like nectar, and they are visited and likely pollinated by various insects, much like flowering plants, in addition to utilizing wind pollination. These parallels highlight the complex, sometimes convergent paths of plant evolution.
Okay, so to quickly recap what we've unpacked today and the incredible journey these plants have taken, it's been quite a tour.
It really has. We've seen the seed emerge as a truly pivotal evolutionary innovation, tracing its development through those 7 key steps, each building upon the last to create a protected, self-sustaining embryo package. We identify the progymnosperms as the likely ancestral link, highlighting how crucial innovations like the bifacial vascular cambium, which produces wood and secondary phloem, actually predated the seed itself.
Right, wood came first.
Then we explored the 4 diverse phyla of living gymnosperms: Coniferophyta, cycadeoids, ginkgophyta, and Nettophyta, and saw how each group represents a unique branch of this ancient lineage, with some phylogenetic mysteries, especially around the nettophytes, still being actively researched. We discussed their diverse reproductive strategies, particularly the fascinating evolution of the pollen tube, from its possibly initial nutrient-absorbing role in groups like cycads and ginkgo, to its sophisticated function in directly delivering non-motile sperm in conifers and nettophytes, completely eliminating the reliance on external water for fertilization.
A major step onto dry land.
Absolutely. And we delved into the unique adaptations and life cycles of each group, from the towering redwoods and the lengthy 2-year pine cycle, to the ancient insect-pollinated cycads with their swimming sperm, the incredibly resilient living fossil ginkgo, and the truly bizarre, almost angiosperm-mimicking Welwitschia and its nettophyte relatives.
And here's a thought for you to chew on as you go about your day. We've seen how gymnosperms perfected the seed and dominated the earth for millions of years, building the earliest immense forests. They were true innovators. But despite their amazing adaptations, their incredible resilience, and their ancient lineage, the next great plant innovation, the flower, would lead to an even more explosive diversification with the angiosperms, which now dominate most landscapes. So the question is, what unique advantages or niches do these magnificent naked seed plants possess that allow them to continue to thrive and even dominate vast regions, standing strong alongside the world of flowering plants in so many ecosystems today? From the Deep Dive team, thanks for joining us on this exploration of botanical ingenuity.